In this episode, chemistry and biochemistry professor Dr. Trushar Patel explores how not to get viral, understanding the communication between viruses and humans. Dr. Patel's research program is timely due to recent global viral outbreaks and the fact that there's often a lack of available treatments or a failure of currently available drugs designed to target viral components. In this public professor series talk, Trushar will provide an overview of human viral communications and discuss some of his recent work.
Namaste and good evening, everybody. And thank you for joining me for tonight's public professor talk, virtually, of course. And we're going to learn some things about how viruses communicate with humans and how do they actually evade our systems and what we can learn in terms of basic research and so on. So before we proceed, let's assume that this is the SARS coronavirus, which we all know thanks to this pandemic. And the SARS coronavirus has this spike protein that is located on the surface. And when it comes in contact with human cell, which has this ACE2 receptor located on the surface, which you don't see, but the communication between the spike protein, so the spike protein and the human ACE2 receptor that allows the virus to integrate to the membrane and eventually enter to the cell. So this is our cell. And once the virus enters in our cell, the virus RNA, another term that we all know thanks to this pandemic, and I'm going to hear this phrase quite a bit in my talk. So this viral RNA will then communicate with human proteins as shown here. And such communications allow viruses to make thousands of copies. And once they make thousands of copies, they will release themselves out of the cell and they are free to infect neighboring cells, right? So that's how viruses function. They are unwanted guests to our cell and they use every machinery that we are supposed to be using for ourselves. So what we do in our lab is we want to understand how this viral and the human communication work. And understanding this language, understanding this communication will then allow us to develop, when I say us, it's us and other researchers out there, will they allow us to develop this treatment or some sort of therapeutics development procedures so that we can stop this communication and thereby stop viral replication. So that's the long-term goal of our research study. Before I begin and talk about the research program and other aspects, I would like to spend a couple of moments on the most important slide for me in this presentation. So, sorry. I would like to thank my family here and back home, and it's without their support and sacrifices they made, I wouldn't be here. Talking about family, I also like to thank my family here at work, my lab. So it's these incredibly talented, awesome people that I work with, and it's usually their work that I brag about. So thank you guys. I'd also like to thank colleagues at my department and outside. To name a few people in no particular order in my department, I'd like to thank a lot of individuals who helped me out since I came here. Some of the names include AJ, Boris, Uta, Ethan, John, Peter Dibble, of course, Mark, and all these people helped me quite a bit settling down on campus. However, I have also enjoyed incredible amount of support from the research services and from the dean's office. I'm very fortunate to get support from these many individuals on campus. Some of the works that I'm going to present was done in collaboration with Dr. Carla Coffin at the University of Calgary and Dr. Boris Denver on SARS coronavirus. So thank you them, and thanks to a lot of collaborators who have helped me so far. My PhD supervisor is in Nottingham, and then my mentors in Manitoba and Birmingham, and you guys, of course, the taxpayers. It's your money that the government then redirects to the funding agencies, and we, the researcher, write the grant proposals, And if those grant proposals are deemed to be okay and pass all this criteria, then we get the money to do the cool research. So without your support, this wouldn't have happened. So thank you guys. And importantly, I'd also like to mention Catherine Rader and her team. They've been working awesome in terms of organizing this public professor talk. Since I came here, I attended almost all of them. And even though we had this pandemic, they turned it really quickly around. And we can at least continue this kind of program virtually, trying to connect the community and the university together. I was actually contacted January 2020 about this presentation, and not knowing that we will be in the middle of pandemic, I chose this topic. So moving on, as I said before, that this pandemic has been very educational. The community, the researchers, everybody has learned a lot more about the viral systems, the RNA viruses, the vaccine development, the drug development, all different aspects. I don't want to remind ourselves that this is not the first viral pandemic. In fact, we have a lot more pandemics before, although not as devastating as this one. We had the previous SARS coronavirus, we had the previous MERS virus, we had Zika, we had Ebola, we had dengue, we had polio, we had hepatitis, bird flu, H1N1. We had so many pandemics already happening. And the one important message that I would want to give from this slide is that this is likely not the last pandemic many of us are going to see in our lifetime. So what can we do about this? What can we do in terms of being proactive and not being reactive? All the measures that we are seeing at this stage are what we call reactive measures, right? So how do you become proactive? And there are certain things we can do to be proactive. And one of the things I would like to point out is investments in education, investment in research, investment in training, because we need to be prepared for any of the future issues, not just viral pandemic, infectious diseases, antibacterial resistance problem, cancer. We have so many health-related concerns, right? So we need to train professionals now so that we are ready in the next 10, 20, 30, 40 years of cycle. We shouldn't be thinking about the 5 years of political cycle. So we have to come out from this concept of the small scale vision, and we have to develop the long-term vision in terms of investments in education and research. We need to learn a lot from this pandemic. History teaches us a lot. It's a shame that we haven't learned as much as we should have learned from all the previous pandemic, but we need to learn some of the important lessons from this pandemic. We need to find out what we can do to actually move forward in preventing the future pandemic. And for sure, if I have a limited amount of money, I would not invest this money in making certain movie popular that nobody knew about it before, but I would rather invest that money in education. As Nelson Mandela said, education is the most powerful weapon which we can use to change the world. And I think this is absolutely essential time to use that weapon. And so this will be my speech or my pitch to the elected officials and decision makers and policymakers and all of them. But as a community, we also have a social contract. Let's not forget that our job as a community member, our role as a community member in this particular case is absolutely essential. It's very critical. We should not think about me always. We should think about us. Any miscalculated step, any bad movements that one person makes affects a lot of people around us. And so we have to think about this individual, we have to come out of this individuality complex, and we have to think about the community. We need to help each other, we need to support each other, and certainly not go out in these kind of scenarios. As Eleanor Roosevelt said, with freedom comes responsibility. So if I'm the anti-mask protester, or if I'm the anti-freedom protester, or I'm the anti-lockdown protester, or I'm the anti-vaccine protester, I need to ask myself, if I'm doing what I'm doing is because it is affecting my freedom, or if I'm doing what I'm doing is because I'm not being responsible. Because responsibility and freedom are two sides of the same coin, and often we forget that we need to add a little bit more responsibility because freedom comes with responsibility. And so together we need to fight misinformation. We need to encourage each other to do the right things. And as Mahatma Gandhi pointed out, the future depends on what we do today. And it literally depends on what we do today. We had 764 cases in Lethbridge alone today, including many variants. And so if you want to get out of this pandemic, and if you want to prevent future pandemics, there are certain things that we can do. And one of them is respect the authorities and try our best not to increase the cases and get vaccines when we get our time. Those are the few things that we can do to get out quickly from these kind of pandemics. No viral presentation or topic these days would go, even the next few years, would go without mentioning the SARS coronavirus. So until today, all my slides would include how the viral cases are going up all over the world. But I think thanks to the vaccines now, we have to change our mindset a little bit to give a positive spin to the story. And we have to appreciate that how quickly the vaccines have come and how quickly we are vaccinating people all over the world. So just to recap, as of yesterday, we have lost about 207 million lives, unfortunately, due to this SARS coronavirus pandemic. And a lot of this could have been prevented. Let's not forget that. We still have about 21 million active cases, and a lot of places are seeing the third wave, as unfortunately likely we are in Alberta. Now, thanks to a lot of efforts from different bodies, which I want to talk about in the next slide, we do have effective vaccines available. And as of yesterday, 460 million today, this morning, 500 million doses have been administered. And this is yesterday's data. As of today, we have now a vaccination program going on in 140 countries. So this is a great deal. And obviously, some countries are doing better than the other countries. But again, overall, we are moving in the right direction. And because the vaccines were developed so quickly, a lot of us have naturally some sort of hesitation in administering the vaccine. And thanks to this pandemic, everybody knows that usually it takes about 10 to 15 years. So I'm going to walk you through the slide because I realized that it's a little bit hard to see some of the fonts. So as pointed out here in the top chart, it takes about 10 to 15 years, as we learn from this pandemic, to develop the drugs or the vaccine. And in this case, it only took us 1 year. So why? So let's look at this. Let's look at the situation and analyze a few things. Typically, the first step for vaccine or drug development is to identify the target. So what part of this virus we are going to develop our drug or vaccine against? So that's the target identification. And then we have to develop the vaccine molecules or the drug molecules. And then we have to make sure that these things work in a preclinical setting. And that takes about 6 to 8 years of time frame. Now, consider this situation we are in. We are already in pandemic. Scientists knew the only way to come out of this pandemic is to develop vaccines. So what did they do? We already had existing knowledge, thanks to the basic research, about the SARS coronavirus. In fact, the spike protein as a determinant to enter the human cell was already established in 2003 after the first SARS-CoV pandemic. So thanks to this previous knowledge, we knew what to target, how to target. In this pandemic, the government funding agencies, the public-private partnership, the academic researchers across the discipline, industry, they all came together, and that saved a lot more time as well. So overall, we cut down that 6 to 8 years of time to, let's say, about a couple of months, right? So that was a huge time saving. And everybody was working together. We needed to get these vaccines to get out of it. And the second major time-saving step was the clinical trial. So typically, it also takes about 6 to 8 years to complete all this clinical trial. And because we're in pandemic, people work together, different organizations work together to organize this overlapping phase 1 and phase 2 trial. So that truncated down time quite a bit. We also had accelerated processes. And so those 5 years gone down to a couple of months. And after the encouraging results from phase 2 clinical trial, many companies started developing vaccines at risk while they are also doing the phase 3 large-scale clinical trial. And the idea was that if the phase 3 trial works out, then we don't have to wait to make vaccine. We already have the vaccine that we can deploy. And those were some of the time-saving measures that are occurring and have occurred, and therefore we have vaccines in short amount of time. And there was no corner cut. In fact, there are 4 vaccines that were stopped at the phase 2 trial because they were not showing efficacy and safety. And so all these aspects allow us to develop these vaccines very quickly. And I really request all of us to get vaccinated when our time comes, obviously not cutting the line short. So before we move on to some of the scientific details, I do want to give some background of the molecules that we work with. So thanks to pandemic, we all know the molecules such as DNA and the RNA. So DNA is a long chain of molecules that contains 4 building blocks. These are known as adenine, thymine, guanine, and cytosine. And remember these G's, they're pretty cool. They'll come back later. So DNA, typically, the 2 strands come together and they make this spiral structure, right? And this structure is the genetic material that humans and plants and many higher organisms have. DNA contains information to transcribe in another very, very important molecule. And again, thanks to the pandemic, we all know this molecule is called mRNA or messenger RNA. 2 leading vaccine candidates are based on this molecule. So DNA contains information to make mRNA, and then mRNA can be translated to building blocks called proteins, okay? So remember this bit, it will come back a few times. And the second slide that I want to give is the complexity of the nature that biological sciences and life sciences people often have to deal with. So my height is 168 centimeter, and you can see me through eyes using visible light, right? And the eyes are about 2.4 centimeters, so you can see all of these things well. Sometimes you have to wear glasses, but you can see me and other people. How about if you want to see this type of cells that eat bacteria, right? So macrophages, for example. So the macrophages shown here are about 0.0015 centimeters. So we started going down quite a bit in terms of the decimals. Well, how about I want to see bacterial cell itself, 0.0002 centimeter. Okay, we can use the light microscope and we can study these molecules, fine. What if I want to see virus? Or what if I want to see the viral protein? So in these cases, the biochemists, the structural biologists, people like us and many others in the department, we have to use some specialized techniques, and they are called either the synchrotron light source or the cryo-electron microscope. So I'm going to give you some results based on these very cool, awesome techniques as well. Okay, so let's switch gears a bit and let's look at another type of virus that is known as hepatitis B virus. Now, this virus contains DNA as the genetic material. The viruses are pretty cool in the sense that these are the only organisms that can contain DNA and RNA and multiple flavors and variety of these molecules as their genetic material. We only have DNA as our genetic material. Okay. So these hepatitis B viruses are present in almost all parts of the world. Despite the availability of vaccines, we have over 250 million people that are infected chronically. including 250,000 people in our country. This virus is very lethal. Every 40 seconds, approximately, someone dies. My talk is about 54 minutes. You can count how many people will unfortunately die within this time. Hepatitis B virus also increases the risk of liver cancer. So this is very, very interesting but lethal virus. I do not want to give you all the boring details about the life cycle, but I do want to give you some key take-home messages from this slide. So like any other virus, this virus also has proteins on its surface. It recognizes the human protein on the liver cells. It comes to our cell, and then the nucleic acid of this virus, the DNA, will eventually end up in a specialized compartment in our cell called nucleus. Now, nucleus is also the place where our own DNA resides, as shown here. So then this DNA of this virus will eventually make mRNA, viral RNA. This viral RNA will use host machinery, make lots and lots of viruses, get out of the cell, infect other cell. Routine thing for viruses to do. The current treatment targets all different aspects of viral life cycle. However, they do not target the DNA, which is the main genetic material. This is the reason of the lifelong infection in many cases. So often when the patients stop taking treatment, this DNA is still there. It'll eventually make virus and the virus will come back. So when I started my lab, we started to look into this idea of, can we actually target viral DNA? It's one of the very novel, unique ideas that's not been explored in detail before, and this is what we wanted to do. So our first approach was to use bioinformatics data set, where there is this large-scale database shown here, and this database has about 90,000 genomic sequences of different types of hepatitis B virus. We wanted to look for a region that we can target, that has some signatures to do. So when we look at this particular signature here, that has multiple guanines, remember I said the G's are very cool, they'll come back. So this stretch of DNA has multiple G's. That got me really excited. And I'll tell you why. As I said before, the DNA is double-stranded structure like this, right? However, when there are multiple G's present, they can actually fold into a very different type of structure, schematically shown here. So each of this plane of DNA is guanine nucleotide. So this is 1G, 2G, 3G, and 4G. So it will sit like this, 1, 2, 3, and 4 G's together. And then another 4 set of G's will come and sit on top of that. And then another 4 set of G's will come and sit on top of that. So that makes this compact structure rather than the long DNA structure. And that was pretty cool for us to observe just based on this nucleic acid sequence. So this is what we thought. We had no proof at the time. The second exciting point that we stumbled upon is when we analyze different genotypes, loosely called the variants of the hepatitis B genome, we also saw that most of these genotypes, except one, everywhere, this particular G-rich area is present. What does that mean? That means that if we develop any therapeutic against this region, we can actually target all of these variants or genotypes. And the third cool point, exciting point, was the fact that somebody already studied this region in the sense that there's a human protein, there's host protein that talks to this particular area of hepatitis B viral genome, and that is required for a virus to replicate. So we had a lot of exciting stories coming out from just one particular study, which was published a couple of years ago. So I gave a lot of information. Let's take a step back. What just happened? Well, we all know that viruses can't survive without human machinery. And the previous study showed that the region that we are interested in is also the region that human protein is binding to or talking to so that virus can multiply. So our next steps were one, to prove that this guanine or G-rich piece of hepatitis B DNA forms a complex structure versus the long structure. And also, if it does, then can we actually develop some sort of therapeutic molecules against it, right? So those were two next goals. And we use multiple approaches to show and prove the first point, that the region that we are interested in does indeed have a complex structure. The study was actually published online yesterday in one of the leading journals. I'm not going to talk about all the results, but I'll talk about one particular experiment because nature does that experiment every day with us, and that is called light scattering. So stay with me here for a minute. So if you ever wonder, and a lot of you know, why the sky is blue, like shown here, during the daytime. So let's say this is Earth, and this is the sunlight here. So sunlight comes, the visible spectrum of the sunlight comes from the top, and it reaches to us. On their way, they interact with the particles in the upper atmosphere. And this upper atmosphere particle will basically separate the spectrum of the visible spectrum of the light, but we only see the short wavelength blue light. And that's why the color of the sky is bluish during the daytime and the evening. As the day progresses, late in the evening, the sun has a different angle. It's right here at the horizon. The sunlight, the visible light, takes longer period of time to come to Earth. They also have to travel through the lower atmosphere, right? And so all this effect actually filters out all of the different shades of light except this red-orange shade, which is the higher wavelength, right? And that's why we see these beautiful colors in the sky after in the evening. So we use exactly same principle of light scattering that nature does every day. We just tweaked a few things. So what did we change? Well, to begin with, instead of the red light that has about 7,000 angstrom of the wavelength, we used Xrays, which is only about 1.3 angstrom of the wavelength. Why do we change this wavelength? Well, if you want to look at the smaller particles that I showed you earlier on the slide, when you want to go 0.00000 kind of sizes in centimeter, then you have to use smaller wavelengths that can penetrate inside this molecule, and you can see the overall surface, right? So we use these X-rays, and we use very high energy, very, very sharp beam of X-rays. And this is the facility in England called Diamond Light Source. It is engineering and physics marvel. There's a lot of excitement when you go there. It's a complicated piece of machinery, but we don't have to deal with it, thankfully. The beamline scientists do most of this alignment and all the other experiments. We go with our sample, we load our sample here, we collect the data using the detector, and then we use a lot of computational approaches to actually study the shape of these biological molecules. So let's see what happened with that piece of DNA that I was talking about from hepatitis B virus. So we took this piece of DNA that has multiple guanines, and we did the experiment that I showed in the earlier slide. And as you notice here, instead of this double-stranded long structure of DNA, we have a very complex structure of DNA, which the dimension of about 47 angstrom. So it's a very small, very compact. In life sciences, in biochemistry, in biology, we do these experiments which call some sort of mutation experiment. So it may be hard for you to see on the screen, but we just took one of this guanine here, and we flipped that guanine to A, or adenine. And a single change extended the structure from 47 angstrom to 60 angstrom. So that single change led to a longer structure. So that is pretty cool. Why is it cool? Because it actually shows us, along with the other result that we just published yesterday, it actually shows us that these guanines are very critical in helping the DNA to get the shape. So after we did all this experiment and showed that we have this unique DNA structure of hepatitis B, then we can do something about it. The obvious next step was to actually look for therapeutic candidates. So we used a technique called phage display technology. Conceptually, it's really simple. You take a tube, and in the tube, you have a piece of DNA, which is, in our case, the piece of DNA that we are interested in. So that's called the target. And basically, there is a library of about 3 billion possible combination of therapeutics. And so you look for the one or a set of the molecules that can recognize the molecule of your interest. So in this case, we found about 10 different molecules or candidate therapeutics that recognized the DNA that we are after, okay? So that took a couple months, and then at the end, we had some therapeutic candidates. We took them, we modified them. That word apparently is actually very popular. Everybody understands it. So we took the therapeutics, we modified them, and the next task was to actually produce these therapeutics. Now, therapeutics production can be very challenging and very expensive. What we wanted is a system that can produce therapeutics cheaply. So I'm from India, the competition is very cutthroat there. The AstraZeneca vaccines that are cheapest ones, most of them are made back home at the Serum Institute. So we know how to deal with the cost and operation infrastructure in a way. It's a joke, by the way. So we wanted to look for a system that is faster, that can give us high amount of therapeutic candidates that we wanted, but also most importantly, something that is quickly adaptable and scalable. So if we ever are going to be successful, we will need partners that can produce liters and liters of this therapeutic. So we have to look for already the approaches that we can scale up and are quickly adaptable. So the therapeutics we had was protein in nature, or is protein in nature. Now, if you remember the slide that I said DNA contains information to make messenger RNA, and the messenger RNA then can be used to make protein. So in this case, we have this protein therapeutics. So what we did is we reverse engineered the system, and we put a piece of DNA in E. coli, or the bacteria, which in biochemistry and life sciences, people routinely use as a biomanufacturing unit. So we put the piece of DNA to bacteria, grew lots and lots of bacteria, and as these bacteria grow, they also make our therapeutics. And then you purify these therapeutics from that bacteria. So this is exactly what we did. After that, the next obvious goal was to actually figure out whether the modified therapeutics that we made in the lab actually recognize or communicate with the particle that it should communicate. So we designed an experiment, and be with me for a few minutes. I'll walk you through the experiment. So one of the first experiments we did, which gave us this straight line, we took this candidate therapeutics. So this is the straight line right at the top, right there. So we took this candidate therapeutic and we mixed it with a linear piece of DNA, which also has a lot of guanines. The hypothesis was that if our therapeutics are very targeted, very specific, they should not recognize the linear piece of DNA. And indeed, the straight line shows that it did not recognize the linear piece of DNA, which is a great start. Okay, the next test was to take that compact folded piece of hepatitis B DNA and then put it together with our therapeutics and see whether our candidate therapeutic actually recognizes what it should recognize. And this curve going down nicely actually shows that it does recognize, not only it recognizes, it also recognizes with very high affinity, or it's a very strong recognition. So that is also pretty awesome. The caveat though, which is not a major concern at this stage, is that there is a piece of DNA that humans also have, which is not too similar to hepatitis B RNA. hepatitis B DNA, and the candidate therapeutic does show some sort of communication, but this communication is very weak. It's extremely weak compared to the original target, which just sits there, right? So we had a really good start. The next experiment we did was to check their stability. Now, believe it or not, this experiment was actually done last January before we all learned, scientists knew already how important the therapeutic stabilities are, but before the community learned from Pfizer and Moderna storage conditions, right? So we want to make sure that our candidate therapeutics are stable. So we did this experiment at minus 20 degrees and 4 degrees and 20 degrees, and we showed that between day 1 and day 7, there is not enough change in therapeutic storage conditions. So this is also pretty cool. We actually now have data for more than 2 weeks. And so that was a cool start. And the ultimate test again was to see whether they do the job in terms of production of viral production. So we set out another experiment. And again, bear with me. I'm going to walk you through the experiment. So we took liver cells, liver hepatocyte cells, and we infected these cells with hepatitis B virus, okay? And the virus is happily growing in the cell. They use this liver cell as the host. And then we divided this set of infectious cells in 4 different sets. The first set, called placebo, only had saline in there, so that is this PBS. The second set of cells has the currently approved treatment at the concentration that is viable. The third set of cells, which are infected with hepatitis B virus, has our candidate therapeutics. And the fourth set of cells infected with hepatitis B virus has a combination of the approved therapeutic and our candidate therapeutic. So let's look at the results, what we are seeing. So on the y-axis here, we have a log scale. On the x-axis here is the treatment concentration. And as you can see, the intracellular and secreted viral production in placebo group doesn't change. It shouldn't change, right? It doesn't do anything. In the current treatment category, in both cases, as expected, there is a decrease in viral production. Awesome. Would you expect that? It's currently approved treatment. However, if you look at these green circles, the dark green circles, as we give the treatment at about 1 micromolar treatment with our candidate therapeutic, we actually decrease significant production of hepatitis B viral particles inside the cell, as well as secretion of these viral particles outside the cell. And that was the most awesome results that we have seen so far. So that got us really, really excited. And so we have done a lot of other work, and based on all this collective work, we filed for an international patent in Geneva in February. We are currently submitting 4 patents in US, Canada, India, and China. A company was born called Quadramix Biotechnology on 1st of March, and we are currently busy writing grant applications and securing funds to do additional research. And so let's take a pause here. We started to talk about investing in basic research and education and training. And look, what can we do with investments in basic research and training? We can do some cool research. And the idea here is that this kind of research actually helps us preventing and addressing these challenges, right? So to be honest with you guys, my department has extremely talented people compared to me. I'm nobody in the department in terms of the talent scale. There are very smart people in our university as well. We have so much talent on campus. We have so much talent, University of Calgary, University of Alberta, nationwide. We have a talented pool of people which are not funded in terms of the research and education avenues that they want to pursue. But if we change this criteria a little bit, today, president south of the border in the press conference about 6 hours ago, announced that their current investment of science and research is about 0.7% of the GDP, and they would want to increase gradually to 2% within their time in the office. And so we have so much talent in this country, which is clearly underutilized. What we can do with this kind of investment is we can make Canada and Alberta global in terms of our reputation and the recognition. We can attract a lot of funding. We can attract a lot of trainees, and we can train them. We can collaborate with the people, and we can satisfy all the metrics that the government has actually suggested us to do. And this is one small step. I'm going to quickly rush through a few slides on the second project. I talk a lot and I got carried away, so I'm sorry. The second topic that we're going to talk about today is on a group of viruses called flavin viruses. So these viruses contain RNA as their genetic material, similar to SARS coronavirus. Some of you have heard about these viruses called Zika, dengue, encephalitis, West Nile, especially in North America, that infects animals, birds, and humans. And so these are deadly viruses, mostly spread by mosquitoes, and they have now presence all over the world. And they are classified as emerging and re-emerging pathogens, right? And so what we are doing in the lab is working on a very basic concept, which I'll walk you through in a few seconds. But basically, these viruses contain RNA, as I said, as the genetic material. And these RNAs have a start tail and they have a stop tail. So it's a long, flat molecule. Let's say this is the start tail, this is the stop tail. These viral RNA tails come together and they talk to each other. And they make this panhandle circular structure once the end talk to each other. Now, these ends are very critical in terms of viral replication. So what we are trying to understand in our lab is how do these viral tails look like? And we have the systems that we work with that are called encephalitis, Zika, Murray encephalitis, and Powassan, which was identified here in Ontario, Canada. So we want to know how this viral tail looks like. And then we want to study their communication with human protein with a similar concept that this kind of basic research can help us and the other groups to develop future therapeutics. So we're doing a lot of experiments, including that light scattering with Xrays. And you probably won't be able to read the fonts, but what I'm trying to show you here is that we have the similar lengths of genomic sequence or genome. And when you study the shape, as shown here, for example, they all look different. As a comparison point, the Japanese encephalitis virus tail is long, it's curvy compared to the Powassan virus or Murray encephalitis virus. So this is very exciting because it allows us to now move forward to our next task, where we can look inside these molecules and figure out a little bit more detail. So these are already very encouraging results. And similarly, we also study the stop tails of these 4 different viruses. And again, the encephalitis virus is long and curvy. A Zika comes next. And then we have the other 2 viruses. So these are all exciting discoveries from basic research that are allowing us to form the foundation for the next steps that we want to embark on. And remember I said these 2 tails talk to each other, right? So this is the 2 tails coming together. This is for the first time anyone has visualized how these 2 tails look like when they are in close proximity to each other. So these are some exciting developments that are happening in the lab. Now, I did mention that apart from these viral tails by themselves, we are also interested in looking at their communication with human proteins, right? So we work with a type of human proteins called helicases, or what I call loosely zippers and unzippers. So helicases, when they come in contact with viral RNA, which has this complicated folded shape, they would actually shred, like unzipping your coat, to a linear chain. They'll separate the chain. And why this is important? Well, viruses, when they come with complicated folded RNA, they do want to get this linear shape often so that they can recruit other type of human proteins for their replication. So these zippers or unzippers are pretty important. And recently, what we've shown in two different publications in the same journal is that this human protein called DDX3X can bind, so this plot going upwards here and this plot going upwards here. These results show that compared to the control, this particular protein binds to two star tails, encephalitis virus and the Zika virus. Why is that cool? It's cool because now we can study this communication in further detail, and we can envision having a treatment that can treat both virus infections at the same time, the same treatment. So that's why it is very exciting journey that we're going to pursue in the future. And the last but not least, of course, everybody knows the current pandemic and the impact of the current pandemic, the SARS coronavirus 2. So here, we started this work with Dr. Demler's lab last summer. And what we want to do here is we chose a unique it's not unique, but a particular protein from SARS coronavirus because it's conserved across all the coronavirus family. And it communicates with another human protein. And this communication, as usual, is pretty useful, or it's required for viruses to replicate. And so idea here is we want to study how the viral protein and the human protein talk to each other. And I did a lot of work in my postdoc to perform structure-based drug designs in computational tools and high throughput drug screening. So we're going to use those tools to figure out what drugs we can choose or what treatment we can develop that can affect the binding of human and viral protein. And this is a proactive measure because we are envisioning that the candidate therapeutic that we'll develop will not only help us prevent the current SARS-CoV infection, but it will help also treat any future, hopefully future pandemics as well, or future SARS-CoV-2 strain. And to that end, we have determined halfway there the structure of the human protein. And to our surprise, this human protein is very interesting because usually you have only one protein together. In this case, there are 6 different proteins that clump together. So it's hard to show you through the screen, but this is 1, 2, and 3 proteins. And the back end, there are 3 proteins. You also notice that this one has a little bit different information than I gave you before. So before I gave you, let's say, a finger kind of shaped molecules, but not details on my wrinkles. And here you can see all the wrinkles. It's because we changed the instrument and we changed the wavelength. So Xrays, 1.3 angstrom, we went to electrons, that is 0.02 angstrom. And that allows us to even look into the further detail. So that was my last slide. I would end my talk here by recognizing my team. So we have multidisciplinary research program in our lab that involves bioinformatics to cell and molecular biology to biophysics, structural biology, and a lot of computational packages and computational calculations as well. So we have diversity in terms of our research program. Here, DJ, Tyler, Sean, Darren, and Cody are working with RNA viruses and studying human RNA, human protein viral RNA communications. Michael and Angela, I haven't shown the results. They are working on hantavirus system. Simone, Dr. Maurik Bartmalmia, Dr. Vanessa Stephenson, Rado, and Nick, they work on hepatitis B setup project. Dr. Kadir Siddiqui, Rutu, and Mitch are working on another awesome system in human cells. and haven't shown the results. And we also have recruited some more people to join us in the next couple of months. So I do recognize that our lab presents diversity in terms of the research program, language diversity, ethnic diversity, geographical diversity. We satisfy almost all diversity criteria. And I recognize that we do need to improve on the gender diversity, and we are working towards it.